In this video, I'm going to go over the data analysis for cell differentiation paper using mock dataset. This is an Excel file similar to what you see for your class data in the Blackboard folder under assignments. However, the difference is that here you only have 3 groups, while in your actual assignment related Excel sheet, you're going to have anywhere from 6 to 11 groups per section. So you want to make sure you're opening the file related to your own section to analyze. So if you're Monday AM, open the file that says Monday AM. If you're Wednesday, open the file that says Wednesday morning or evening. So you have the appropriate data available. Now, in the Excel file, when you open it, you're going to see 3 separate sheets. The first sheet is going to give you the cell differentiation information. These are absolute number of cells that were at each stage in all your treatment conditions out of a total of about 200 cells that you observed. You will notice some people observed slightly less or slightly more than 200, which is why it is important for us to note that these are not just 200 cells. You have to convert them into percentage before you can compare between the different groups. The other 2 sheets are going to contain the average data for cell measurements, the cell body diameter, and for either projection lengths or projection numbers. You may or may not have this 3rd sheet available depending upon which section you belong to, as some of the sections did not collect projection lengths or the number of projections that are found per cell. You will use the data that is available to you along with data that you collected for your own group. The cell measurements as well as projection length sheets are actually secondary analysis. You first need to present that information for your own group, and then you can analyze the data that is provided to you with the combined class averages if you so choose. However, that is not required for the paper. What is required is all the analysis that I'm about to show for cell differentiation, and in addition to that, you need to show all the data that you have for your own group's measurements for cell body diameter as well as projection analysis, whatever that analysis may be. It may be that you looked at total number of projections per cell, or it may be that you looked at the length of projections on a cell. So whatever it is from your own group is what you're required to show for the paper. For the cell differentiation, however, which is the sheet 1, you need to combine the class data. You need to look at all the data that you have for all the groups and analyze it all together so that you can perform proper statistical analysis between the treatment groups. The statistical analysis that you're going to perform is not between your data compared to the class data, but rather between the treatment groups, that is between baseline versus high serum control versus low serum control versus TMSO and RA. Okay, so we are going to start by looking at the cell differentiation sheet first. To do that, I usually like to make a separate sheet, open up a separate sheet by just clicking on the plus sign. That allows me to do whatever analysis I want to do in a separate area without worrying about accidentally deleting important information from my actual data sheet or moving things around where I don't want them moved. What I will do first is then copy over the data that I'm getting ready to analyze into this new sheet that I've just created. I can do that simply by selecting the data that I want to analyze, copy it over, and paste it into this new sheet. And here I have it. I now have the same data that I had in sheet 1, labeled cell differentiation, copied over into this new sheet that I've created. Now, before I start converting them into percentages, I'm going to go ahead and copy my columns, which contain all the information for the treatments. So I will take column A, copy it over here where I want to use it. So here I have gone ahead and copied all my group, all my treatment information into a new column. I will also take all the groups and copy them over here so that I know how many groups I need to worry about. Now, some of you may have tried to look at this by simply adding up the total number of cells in each and then just averaging them out. That is not correct. You actually need to analyze the data group by group and then combine it for statistical purposes. Otherwise, it would not work appropriately for you. So what I will do now is I'm going to go ahead and start analyzing it by first converting it into percentages. This is absolute number of cells. So here I will start with the baseline and go equals, and I can go ahead and put in a parentheses, select my number of cells from the first column, divided by total number of cells that are present within that sample, and then multiply by 100. So this is going to give me percentage of cells that are undifferentiated in group 1 sample in my baseline. When I do that, I'm able to now go ahead and get that percentage. I can simply drag this over and it will automatically pass on to all my other groups cells and calculate for me percentage of cells that are undifferentiated in each one of my group samples. I can similarly do the same process over for all my other stages and get the number of cells from here and change them to percentage of cells that are at stage 1, 2, or 3 in each one of my group samples appropriately. Okay, so now I've gone ahead and I've added in the formulas for each one of my stages. I can go ahead and drag this whole section over to as many groups as I have, and it will go ahead and automatically select this information for me. I can next just copy this over and take it to the next section, which is the high serum control, and paste the formulas, and it should automatically calculate the appropriate cells for me. As you can check here, double check to make sure it is doing the right thing. So D17 divided by D18 multiplied by 100. So here I will go ahead and now paste the formulas throughout my sheet so I can get all my numbers appropriately.